for all the sections of the analysis. Today we will be working mainly with 4 sections: the RNA-seq section, and the data mining section, the supervised analysis section, unsupervised analysis section, and utilities section. All sections have similar interface, so let's go to RNA-seq section to get a hand of how to make the pipelines. First, what you are asked to do is to fill out the type of data that you are going to upload. So type of reference genome refers to the reference genome you are going to align everything onto. R model genome GTF means that the genome and the GTF file, annotation file, are already in our repository. Usually we will be using, today we will be using only this option. And here are the organisms for this genome. And this is of course needed only if you are uploading from our repository. So these are all the available organisms. Another option might be R genome, and that means that you are uploading your own file, customized genome. You can also be uploading both the genome and GTF file, customized genome and GTF file. For these 2 options, the organism will not be present. Today, we will only be using the R model genome GTF. We will be using the human genome, the newest human genome, this one. For the PDX data set, we will be using the concatenated genome of human and mouse. The data format we will be working with today is FASTQ. Other formats are also possible. The most popular formats of NGS data are FASTQ and FASTA formats. You can be also working with alignment data, already aligned data with SAM or BAM files, and you can be using, of course, simulations for which you don't need any data, previous data available at all. The NGS data comes in 2 formats, single-end and pair-end, and this should also be identified here in this before you actually upload your data. So we will be using the pair-end data today. After you have chosen what is needed here, you upload your file. We will be using today SVL links, bulk upload of many files, many SVL links together today. But of course, you can upload your own data from your personal computer or HTTP. After you upload your data, you are asked to define the groups. Dr. Galetinko will go into how you choose how many groups you need. So I will not go into this now. So I will do cancel no groups. And then I am ready to actually prepare my pipeline. T-bio.info platform. So when you are ready to prepare the pipeline, you will notice that some buttons appear in a bright black lit up section and others are gray. Only the black ones are you are able to press at each time. This was made for convenience because we think that sometimes choosing the algorithms is not so simple. So we wanted to help the user move along to creating this pipeline. Also, each time you press a button, you will see a pop-up window where the algorithm is explained, and sometimes you will also have to choose parameters. So let's begin with the start. So I press start, the pop-up window comes up with explanation right here of the algorithm, in my case, just what is RNA sequencing, and okay. Sometimes on this side, you will see parameters of the algorithm. Now, when I chose start, I have few options. As you can see, some of the algorithms appear in bright colors, which means that I can actually choose them, and some are gray, which means I cannot choose them. Let me try top hat algorithm. And again, pop-up window with an explanation, no parameters here. So I press OK. When I am done, I simply press End and save my pipeline. I can choose a name of the pipeline, let's say test. This is just for you, and you can run your pipeline. When the pipeline is finished, there are two options. You can always go to My Pipelines and check the status of the pipeline. You can also receive an email. You also receive an email with notification of problems of the pipeline or that your pipeline has been finished. We will start with an overview of transcriptomic data and initial analysis. And we will then proceed to unsupervised analysis tools for analyzing expression table, including annotation and pathway analysis. Then we will have a section on unsupervised tools, and we hope to finish with the time for practical independent analysis of data when the team will be there to help you and support you. A reminder, throughout the workshop, we will have frequent breaks when you can ask Dr. Galatin questions or ask for assistance from our team. Let us know if you need such assistance right now through chat, or if you're in the audience, please ask Sahil. Let us take now 1 minute break to make sure that everyone has all the files ready and the platform open, and we will welcome Dr. Galatin. Thank you.
To our online participants, is everything going good? If there are some issues, you can probably ping us on the chat box, or you can drop us an email. Now proceeding to the next one. Now it's to Dr. Galantango to start up with his things. Dr. Galantango, can you please unmute yourself? Yeah, thank you.
Okay. Okay. So before we start, please let us make sure that we all have an access to the server.t-bio.info platform, and we all have the set of files we'll need. So we all have an access primarily to the screen with sections we'll be working with, and we all have the set of files out of which our main subfolder will be main files. The additional thing we'll need is any program for table processing, for example, conventional Microsoft Excel, OpenOffice, or something like that. So if it is okay with these points, let us start with working with RNA sequencing datasets just from the beginning. So in order to understand what is the processing of RNA sequencing data, we first need to understand the scheme that is used at a wet lab level to process all these data, to prepare FASTQ files. So initially, we start with a genome, and as you know perfectly well, certain sections of genome code for proteins. So these sections are called genes. Proteins are not produced directly from genes, but there's an intermediate step, the production of messenger RNAs, and all the cells in an organism have the same genes, but these genes have different expression levels in different tissues, in different organs, in different cells. And so the expression level, so ideally we want to measure the rate of production of proteins, but technically it's more untrivial and it's more expensive to look at proteins. However, it also can be done, for example, via mass spec experiments. Yet, looking at all the transcripts at the messenger RNA level is relatively straightforward and relatively cheap, and from the low detailed viewpoint, the situation here is the following. All messenger RNAs are simply cut randomly into short pieces. Initially, we do not know the origin for each piece, but in any way, more transcripts corresponding to a gene we have in a sample, more short segments originated from these transcripts are observed in the whole stack of these short sequences. These short sequences are sometimes called reads. So in order to analyze the expression levels, in order to analyze the activity of various genes, all we need to do is to map these reads back to transcripts. And this task is feasible as for model organisms, and in particular for humans, we know the nucleotide sequences for all the genomes, so we simply map the reads read by a sequencer machine back to transcripts, and if there are many reads mapped on a gene, or mapped on, let it be mapped on a gene, then the expression level of this gene is high. If there are only few reads mapped on a gene, the expression level is low, and for many genes there are no reads mapped on the gene. It means that the expression level of this gene is 0 or little. However, if we look deeper into the details, the process is just the following. It indeed begins with shattering all the transcripts, all messenger RNAs in a sample, for example, via ultrasound, or there are various other technologies. But then there is an intermediate step required for sequencer machine to be able to capture these short segments, these reads. So adapters are ligated generally from both sides of the reads. These are just the same sequences that are ligated, well, roughly speaking, to the beginning and to the end of all the reads. Then so-called PCR amplification step is performed when for each segment with adapters ligated, multiple copies are produced. At each iteration of this PCR amplification, the number of copies is doubled. So, for example, if we have 6 rounds of PCR amplification, the number of copies will be 2 power 6, that is 64. If we have, for example, only 2 rounds of PCR amplification, we get only 4 copies for each segment. And so after this step, there are multiple copies for each initial short sequence. For PCR amplification, there is no guarantee that the efficiency of this stage will be equivalent for all short reads. So for some short reads, there will be more copies. For some short reads, there will be less copies in comparison with the number we expect. Finally, at the stage of reading all these short sequences, all these reads by sequencing machine, not all short segments are taken, but only a subset is in fact randomly selected from the whole totality of what we have in a sample. And if we perform multiple steps of PCR amplification, then the probability of reading several copies of the same initial segment becomes rather high. However, if the initial amount of RNA material is sufficient, if the number of steps in PCR amplification is relatively small, well, for example, 6, 7, 5, then the number of these artificial PCR duplicates, so the copies read originated from the same short segment, is relatively low. So these short segments are read by a sequencing machine, and then the process is performed as we initially discussed. All these reads are mapped on gene sequences, and expression levels are estimated. So this technical pipeline provides us and gives us an insight into how we should organize a pipeline for analyzing the results of sequencing. What models should we use when we have FASTQ or FASTF files originated from a sequencing machine? So at a hardware level, or better to say, at a wet lab level, the first step was a ligation of adapters. So a standard pipeline for the analysis, for the estimation of expression levels, starts with removing these adapters. Additionally, sometimes it is a good idea to trim ends of reads, as these ends generally have lower quality. Then, as we might have PCR duplicates, so multiple copies of the same initial segment read, we can remove these PCR duplicates simply by removing reads that are identical. Then we can perform mapping, but there are multiple options for mapping. It can be performed either on transcriptome, so unknown transcripts. In this case, we'll quantify only expression levels of known genes. Another option is to perform mapping on a genome and to at least try to identify novel transcripts, that is, new isoforms for already known genes or even completely new genes. Definitely, high throughput methods, including RNA sequencing, give us an ability to do it, but an independent validation will be required to be sure that identified transcripts really exist. So it's not just a consequence of analyzing too large amount of data. So combined strategies are also possible. A slight problem with this analysis is that it's really time-consuming. The initial size of files is several gigabytes, up to dozens of gigabytes, so even uploading these files takes time. Processing also takes time. However, creation of this pipeline is fast, and there's generally no problem with time taken for processing for quantification of expression level, as wet lab stage takes even much longer, and sample preparation is also a relatively long task. So in order to have a training of creation a pipeline aimed at analyzing the expression levels, for example, of known genes, let us try to log into the platform and see how it can be done. We won't start this pipeline as it takes time, too much time to be finished in the frames of our workshop, but still we'll create it. So as you saw in the introduction, we can select the section RNA-seq or chip-seq data, so the top left section, and start with setting main options. The main options for the model organism include the organism itself. For example, for Homo sapiens, we select Homo sapiens, one of the genome assemblies. The format of input files, generally, if we start with raw data, it's FASTQ, and the type of reads, single-end or pair-end reads. Then we proceed to uploading files. As we initially discussed, the normal size of FASTQ files or FASTA files is really large, but some files are already uploaded to the platform, and in this case, to avoid multiple uploading of the same files, we can use bulk upload for just uploading a file via link to its location in a server. So we can select an option, bulk upload, and from the file list we have, specifically from the folder main files, we can select a link to files that are already present. For example, links to samples will give us a link to 2 samples we have. So we open these files and continue. We open these files and go on. So going on is just pressing define groups. In this first analysis, we won't perform any deeper analysis, differential expression analysis, for example. We'll just quantify the expression level of genes. That is why we simply skip all additional steps and start creating the pipeline. So again, this pipeline just follows the logic of the wet lab preparation of the sequencing data. So after selecting start and confirming it by pressing OK, we select a module that performs adapter removal and potential additional trimming of low quality ends of reads. Then we select a module for PCR duplicates cleaning. Then we select a module for mapping on transcriptome if we want to have simply expression levels of known genes or isoforms. There are at least 2 options, the standard bowtie 2 and also standard mapper BWA. So we can select any. But after mapping, we additionally need to go from read counts to expression level quantification. So we'll need to discuss what are the standard units for the quantification of expression levels. In any way, this job can be performed by the RSM module. This job can be performed by the RSM model that's here called RSM expression table, and the desired expression units, fragments per kilobase per million, expected counts on transcripts per million, can be selected as a job option. So that is all. All we need to do next is to finish our pipeline, give it a name, for example, expression level quantification, but you can give any name you like. And if we wanted to start this pipeline, we could simply press run pipeline, but as I said, there's no reason doing it now. However, later, definitely you can check the result. Okay. So let us check whether there are any problem at this stage or everything goes fine. So if everything goes fine, we go further and discuss our options at creating these pipelines. Available literature shows that trimming low quality ends and even trimming adapters really improves the quality of expression level quantification, but this improvement is moderate. Moreover, removing of PCR duplicates is sometimes not desirable, as in many cases these PCR duplicates are not artificial, but there are natural identical reads. For example, in case of analyzing short sequencing, microRNAs as an example, identical reads emerge from biological origin of the analysis. So in some cases, PCR duplicates removal can even introduce bias to the result. In case if we want to at least try to look at novel transcripts, we simply add several modules to our pipelines. These modules additionally perform mapping on genome, gather exons into potentially new isoforms or even potentially new genes, and then combine in the set of initially known genes with the set of genes that are identified by the pipeline. So technically and algorithmically, it's not a simple task, but from the point of view of a user, the difference between these pipelines is just an addition of several modules. Independently of selecting or not selecting some preprocessing steps, we need to discuss what are the standard units for the quantification of genes or isoforms expression levels. So initially, we started with an idea that more reads we have mapped on a gene, the higher the expression level of this gene is. But let us consider the following two facts. First, imagine we have two messenger RNAs. The first corresponds to gene A and the second corresponds to gene B. And imagine that gene A is much longer than gene B. So after shattering into small segments, this one messenger RNA will give essentially more reads, essentially more segments in comparison with messenger RNA, in comparison with transcript that corresponds to gene B. So initially, we have one messenger RNA for both genes, but after shattering, we have essentially different number of reads, essentially different number of short segments associated with these genes. So if we want to perform scaling that takes this fact into consideration, we should use not just the number of reads mapped on a gene, but number of genes scaled by the gene length. So standardly, here gene length is measured in kilobases. Another fact is the following. Imagine that we have one sample that is sequenced twice, and in one case, the number of reads sequenced is, for example, 2 times higher than in the other case. So, in other words, one sequencing is twice deeper in comparison with the other one. Again, we deal with the same expressions of the same genes. We simply change experimental conditions and get approximately 2-fold difference in the number of genes mapped on a gene, just because in one of the experiments, the total number of genes was 2 times higher. So in order to perform scaling that takes this fact into consideration, the previous expression is additionally divided into the total number of mapped reads. So these two scaling, a scaling by a gene length and a scaling by a total number of mapped reads, is an attempt to reduce the dependence of the expression level we finally have on experimental conditions and on gene length, to have something uniform with respect to genes and experiments. So this is one of the very popular measures for measuring expression levels, fragments per kilobase per million map reads. Another unit for expression level quantification is very similar, and for each sample, this second measure is simply proportional to the first one. The complete number of fragments per million for each sample is just scaled in such a way that the sum will be 1 million. So the new unit we have is called transcripts per million. What is the logic behind these new units? We can define it in a slightly different way. New units can be defined as follows. Let us consider an expression that is equal to the number of reads mapped on a gene multiplied by mean length of a read and divided by gene length. So what is the meaning of this expression? So having a product of number of reads mapped on a gene and read mean length, we have the number of nucleotides, in fact, total number of nucleotides originated read sequenced from a gene. After division by gene length, we have an approximation of the number of transcripts sequenced associated with these genes. So number of reads mapped on a gene multiplied by read mean length and divided by gene length is simply an approximation of the total number of transcripts sequenced for this gene. If we divide this expression by the total number, by the sum of all these expressions over all genes and multiple, for example, by 1 million, we'll have the approximate number of transcripts associated with this gene in every million transcripts we have in our sample. So that is why Transcript per million is a unit that has a natural interpretation, for example, unlike fragments per kilobase per million mapreads unit. Another fact we should take into consideration when working with sequencing data is that in case of any standard unit, counts, FPKM, TPA, in many cases, it's a good idea to go from initial linear scale to logarithmic scale, to log scale, for example, by calculating a logarithm. Standardly, this logarithm has base 2, and one of the reasons for doing so is just a statistical one. For many genes, expression of these genes, expression levels of these genes have log normal distribution. So after taking logarithm, we obtain normal distribution that is very good from the point of view of statistical analysis. So parametric testing is applicable for this case. Another reason is that relative difference is more meaningful in the biological case than the absolute difference. For example, if we say that the difference in expression levels is 100 FPKMs or TPMs, the information contained in this sentence is relatively low. If we say that the difference is twofold, threefold, fourfold, we give more information to a reader about an expression change of this gene. And these relative changes are quantified very naturally in the logarithmic scale. So twofold change is simply a difference in 1 unit in the logarithmic scale in case if the base is 2. Fourfold difference equals 2 equals difference equal to 2 in the logarithmic scale, and so on. The problem here is that the logarithm of 0 is undefined, and for many genes, their expression level quantification is simply 0, so we can't perform log scaling directly. To avoid this problem, a small shift is added. In many cases, this shift simply equals 1. It does not affect quantification of expression levels of genes with relatively high expression or even moderate expressed genes, but it saves us in case of genes having very low expression or 0 level expression. Please keep in mind that genes with low level expression are hard for the analysis, as their expression level is measured unprecisely by almost any type of the analysis, including RNA sequencing. So if the expression level of a gene is really low, for example 0, we should keep in mind that it's just an approximation of an expression level with potentially high error, high just technical error. So what we have after the pipeline that provides us expression levels of all known genes. Let us just take a look and compare the effect of various preprocessing steps. We'll do it briefly and then go on with more hands-on experience. So in the files you have, there's also a folder with some output files. So the subfolder res output files. And there's a subfolder RNA-seq comparison. In the subfolder, you can find a file. With results of pipeline we initially prepared together, standard preprocessing, which includes both adapters trimming and PCR duplicates removal. short preprocessing, no PCR duplicates removal, and just no preprocessing. Everything starts with mapping on known transcripts. And here, units are logged to, here units are just, log FPKM with the base equal to 2 and shift 1. So what do we have here? What do we have after this comparison is the fact that generally, indeed, preprocessing scheme has very minor effect on the results. However, if we completely avoid preprocessing, this effect is higher than in case if we avoid just one step of preprocessing. But if we look at certain individual transcripts, the difference emerged from different schemes of preprocessing is really high. So please, again, keep into mind that it is a logarithmic scale. So, for example, a difference for a transcript for dot I point at is more than twofold. So if we change our preprocessing scheme, we get very different results. That is why for the majority of high throughput methods, including RNA sequencing, additional wet lab validation is highly desirable if our analysis is aimed at the identification of a small number of individual transcripts. So in any case, this pipeline that is technically very simple just provides us with a huge number of various expression levels. In case if we also try to identify novel transcripts, the length of the set of expression levels is even more high, as we get expression levels for potential new isoforms or potential new genes, additionally to the expression levels for initially known isoforms and genes. So the main question is what to do next with all these files. What information can be obtained further from all these expression levels, which are infeasible for the analysis with just eyes, which are infeasible for manual analysis. So let us currently have a short break, discuss the questions, and then go on with the analysis of what can be done with all this data, how this data can be analyzed. So currently, in this break, please feel free to ask questions.
Thank you, Dr. Valentenko. And all the online users, if there is any question, any doubts, please feel free to chat with us on the chat box. We are probably having a break for like 2 minutes, just to make sure that everyone is on the same page. And to our live audience, do we have any questions?
Okay, so probably let us try to go further in order to have a bit longer next break. So while we still have our attention, let us briefly discuss standard ways for differential expression analysis and touch pathway and gene set enrichment analysis. So in many cases, samples can be initially divided into several groups. In the simplest case, just in 2 groups. And the idea that lies behind the analysis is to identify genes that have essentially different expression levels in these 2 groups. For example, genes with high expression level in one group and low expression level in another group. What are the main characteristics of this differential expression? What are the main characteristics that should be kept in mind when thinking about the differential expression analysis. Naturally, the first characteristic is fold change, so the difference between mean expression levels in the first group and in the second group. So it's very natural to say that the expression is sufficiently different if, for example, the difference between means is at least twofold. However, please consider the example we have in this slide in the right. Intuitively, we understand that there is no differential expression if blue dots visualize samples from the first group and red dots visualize samples from the other group. At the lower figure, at the lower plot, we see clear differential expression. We see that the expression level of a gene is higher in blue group in comparison with the red group. The interesting thing here is that the difference between mean expression levels for the top figure and for the bottom figure are identical. So in both figures, blue group has mean expression level twofold higher in comparison with the red group. Here, I pay our attention to the fact that when we speak about mean expression level, standardly we speak about standard conventional arithmetic mean in logarithmic scale, and it corresponds to geometric mean in initial linear scale. But here the numbers are associated with the logarithmic scale. So that is why these pictures show that evidently we can't look only at the difference between means, only at the difference between mean expression levels to estimate the differential expression rate for a gene. We additionally should add some statistical significance that, as an example, we'll compare the difference between means with variation of expression levels inside these groups, the statistics that lies just behind a conventional student's t-test. So fold change is always supplied by statistical significance level, probability value, or p-value. What's the meaning of a p-value? P-value is simply a probability of observing the difference we have, or even higher difference, in case when in reality there is no difference in expression of these genes in two groups. So, for example, if p-value equals 0.01, that is 1%, it means that the probability that we are mistaken telling that this gene is differentially expressed is simply 1%, 1 out of 100. But here we simultaneously test multiple hypotheses. We simultaneously consider multiple genes. So imagine the following situation. We have one coin, we drop this coin 10 times, and this coin falls the same side up for all 10 times. Does it look suspicious? Probably it does, as the probability of having the same side for a coin up in 10 throws is really low. But if we ask several thousand or say million of people, do this experiment, take a coin, throw it for 10 times and observe the result, definitely we'll have many people with a coin that fell all 10 times up the same side. It does not mean that all these people have, well, non-standard coins. It simply means that the number of people, number of tested hypotheses is so large that even events with low probability are observed. So we need to correct this standard p-value and a correction should be aimed at considering this fact of simultaneous testing of multiple hypotheses. That is why standardly these value is supported by so-called adjusted p-value that has different names, slightly different statistical meanings, and sometimes it is called adjusted p-value, sometimes it is called q-value, sometimes it is called, for example, false discovery rate. So false discovery rate means, for example, false discovery rate of 5%, 0.05, means that if we take all the genes, if we take all the hypotheses with if they are lower than 0.05, then only about 5% of genes in this list would be false discoveries. So there would be some false discoveries here, but the majority discoveries will be correct. Additionally, standardly, people also look at mean expression level of gene where working is, and a quotient is required for low expressed genes, as we initially discussed that generally the precision of estimation of expression level is much lower for low expressed genes in comparison with genes with high expression. So let us try to see how differential expression analysis, at least at the level of unadjusted p-value, of standard p-value, can be performed manually, as it is really simple. So imagine we have an output of a pipeline that simply measured expression levels of genes. So let us take the file from the folder main files for differential expression fpkm, well, and try to open it in an Excel or a similar tool just to have a normal visualization. So what do we have here? This is an example with two groups associated with breast cancer cell lines. The first 6 samples, as you see from the titles, are associated with one subtype of breast cancer, and the second pack of samples, 5 samples, is associated with so-called normal-like subtype. And here we have about 60,000 genes. The majority of these genes simply have 0 expression, but the question is how to identify differentially expressed genes quickly. Units that are used here for the quantification of expression levels are fragments per kilobase per million, so FPKM. So what is the standard pipeline, the standard set of steps for differential expression analysis if we limit ourselves with very simple statistics? Again, first, standardly, we go from linear scale to logarithmic scale to be able to apply various parametric statistical models, for example, t-test. So let's try to do it together just to see that the task is really simple. So first, let us transfer all this data to logarithmic scale. It can be easily done in multiple tools, in R, for example, if you like coding, but even table processing tools such as Excel give us everything we need. I just copy a title to see the number of columns in a more simple way, but the first thing I do is I'm going to logarithmic scale. I say that here I need to compute logarithm with base 2. In certain cases, Semicolon is used to separate the first argument and a base in Excel. In certain systems, it is comma, so definitely you know what's the correct way for giving formulas in your system. But again, sometimes it's a semicolon put here, sometimes it's comma. Then we simply transfer this formula to all our other cells. In order to do it, for example, in a standard Excel, I can press on the bottom right corner and move the mouse to the right, and then by again double clicking this corner, I put this formula to all the cells in our file. But in many cases we see an undefined value here. The reason is that, as we discussed initially, the logarithm of 0 is undefined, and standardly when we go to the logarithmic scale, we add a small shift to the argument, to the expression level measured in FPKMs, for example. For example, let's shift equal, well, classical value 1. And just transfer this formula to all the cells. So I'll add colors to see difference, to see groups more visible. And then what are the main characteristics we'll use? As we discussed, it might be interesting to look at mean expression levels to be cautious with genes with low expression level. So let us just add mean expression level for the group 1 for Claudin low, and mean for group 2. So it can be done with a function average in a classical system. Here I've got just localization, so the standard formula is just average, and you show which cells should be considered here. So here's the formula. I need here to change the function, just as currently I have computer with a relatively strange localization. And next I add a similar mean logarithmic mean expression level, so average. For the second group, again, in my case, I simply have to replace average with a same command in my current localization. So here we see just mean values. Having mean values, we can compute log fold change immediately. Log fold change. Sorry. Log fold change is a simple difference between mean levels in logarithmic scale. So what's the meaning of this logarithmic fold change in a standard scale? For example, log fold change equal to -1 means that the expression reduced twofold. Log fold change equal to 2, simply to 2, means that the growth of expression level was 4. If log fold change is 2.3, it means that the growth in the initial linear scale is 2 power 2.3. So here we got our fold changes in logarithmic scale. And in order to get p-value in a standard t-test case, we can use again a standard Excel function, as p-values are provided by a standard t-test function. Again, in my localization, I have the same function with a slightly different name, with a bad C. And here we need to specify the first group of samples, the second group of samples, the second group of samples, p-value type one-tailed or two-tailed, standardly it is two-tailed, and finally some statistical parameter related to our assumption about variation of two samples. So here we have at least two applicable options, that arguments 2 and 3. So first I'll select 2, and then I'll just show you I'll select 3. So here we have our p-values. Please pay your attention to the fact that if we use a slightly different statistical model, we'll have sometimes similar but really different p-values here. For example, in one statistical model, we have p-value for the third line equal to 0.20. In the case of another statistics, just a moment. It became 0.19 and so on. These values are really similar. This difference is insignificant in this case, but we just see that application of different statistical methods gives us different p-values. And that is why it's really important to specify the type of the differential expression analysis that is used, as really different approaches can provide us different results with slightly different sets of differentially expressed genes. So what we have here is logarithmic fold change and p-value for each gene. And in order, for example, to get genes with that are conventionally called upregulated, so differentially expressed genes that are upregulated, we need to set thresholds, for example, greater for logarithmic fold change. So we select only genes for which logarithmic fold change is at least 1, and a conventional threshold for p-value is 0.05. But again, I pay you attention to the fact that standardly adjusted p-values are used, not initial p-values. Here we just avoided a step of computation of adjusted p-values. So the thresholds 0.05 for p-value, or better, much better, adjusted p-value, for example, FDR, and at least two-fold change that is equivalent to 1 in logarithmic scale are in fact softest thresholds that are generally used. In some types of the analysis, for example, if we analyze microRNAs, even softer thresholds are applied sometimes, for example, softer thresholds on fold change, but these cases are rare. And so, doing so, we have a list of genes that are upregulated. It means that for the second condition, they have statistically significant significantly high expression level in comparison with the first condition. If we want to have down-regulated genes, we simply select genes with sufficiently negative logarithmic threshold, for example, log-fold change lower than -1. So if there are too many genes in this list, then we can apply harder thresholds. The reason for having such a great number of genes here, the primary reason, is that here we simply dealt with unadjusted p-values that really increased the number of genes. However, these statistics we used here is not, well, the most powerful one in a way. Statistical models based on negative binomial distributions are used much in many cases and provide in many cases better results in comparison with these t-tests we used. And in order to get differentially expressed genes originated from more complicated statistical analysis, it is much more simple to avoid performing all the steps just by hands. We can prepare a pipeline that does all the job. We can prepare a pipeline that does all the jobs, and let us see how it can be done. Let us again go to the platform. It will be one of our, well, probably last pipeline that won't be run today. The rest of the pipelines will be launched and we'll see their results. So let us create a pipeline for differential expression analysis that uses much more complicated statistics in comparison with the student's t-test. So again, we go to the RNA sequencing section. Again, we set type of reads to pair end and select Homo sapiens and as an organism. And in this case, again, to avoid uploading huge files, we use bulk uploading, but currently select links to 11 samples you saw in the previous Excel file. So the file links CLU versus N-like. And go on. The lower 5 samples here are associated with the second group, normal-like, and the first 6 samples are associated with the first group. So in order to perform differential expression analysis, we need to specify which samples are associated with each condition. So we simply drag and drop samples. I said that the lower 5 are associated with the second condition. We drag and drop samples to the boxes associated with our groups. We set the group, so here we can next press continue. In this simple analysis, we don't need a trivial contrast, so we simply submit it. And as we again start with raw files, initially we can perform the same quantification of expression levels. I just recreate the pipeline we initially created. But at the last step, we add a module for differential expression analysis. For example, HR or DSEC2, both of these modules are based on negative binomial distribution models, but details are different. So we can add any of these two modules. So here is the pipeline that after pressing end would be ready for launching. In reality, These more complicated models for the analysis of differential expression deal not with non-trivial units of expression levels. In reality, they all take simple counts from RSM and perform post-processing, perform quantification in their internal units inside these jobs, inside these models. So again, as this pipeline starts with initial raw FASTQ data, it's rather slow, but let us look at the output of this pipeline. In order to do it, again, we might go to the subfolder res output files. Here, subfolder differential expression, and we have outputs for both HR analysis type and DSEC2 analysis type. It's simpler to open these files again in table processing environment, for example, Excel, and to see just the type of the output we have here. For both cases, the main columns are fold change or log fold change, which tells us the difference between mean levels, statistical significance, here adjusted p-value, and the basic, the base expression level. It is a mean expression level in the first group or mean expression level in all the compared samples, and depends on the tool. So in any case, we have three main columns. So here they are for DSEC2 output. Just a similar thing is for HR output. So logarithmic fold change, statistical significance, and in a way mean expression level, so base level that can be used to carefully examine genes with lower expression. So having the complete table, we can simply look only at genes with the highest level of differential expression, with the highest absolute values of fold changes, and with the lowest with values of adjusted p-values, for example, values of FDR as close to 0 as possible. But what we can do having these lists? So let us, for example, consider an example that we identified certain genes of interest we want to look at further. So just a simple, to take it as a simple example, in the folder with the main files, you have, for example, file cell lines 15 genes, which contains kind of information. Currently, it's even not important what this information is for 15 genes. Another option is we could simply set thresholds and get genes with the highest level of differential expression. In the case of Tauber.bio.info platform, and in the case of many other platforms, the primary type of the identifier used is ensemble IDs, so it's relatively hard to understand the function of a gene, to understand what's going on with a gene using just an ensemble ID. So there are only few people who remember ensemble IDs for many genes. Gene symbols or gene ontology terms are much more informative, but in many tables we do not have this information. But adding information about gene names, gene ontology terms is really simple, and it can be performed not simply by hands, but again using the platform. So let us just create a pipeline and run it to see how simple it is. So let us go to the areas of the analysis. areas of the analysis. Then go to the utilities section. Then go to the utilities section. Upload a file we want to work with. In this case, we perform normal uploading of the file, so it's a standard case. From the subfolder main files, for example, let us upload this file, cell lines 15 genes. And simply continue. So if we want to simply annotate genes by adding their gene ontology terms, gene symbols, and something like that, we create a pipeline with just one module, annotation. Annotation. So in our case, the default options will do. The main options here are whether to output gene ontology categories or not, whether the table you use as an input has a header, and the number of columns in which ensemble IDs are located. Other options are commented, so comments or simple pop-up helps can be viewed after pointing to the isoforms near each parameter. So we confirm using default parameters, give a name to this pipeline, for example, simple annotation, and run this pipeline. So it may take up to several seconds or even half a minute to start. So generally it's a simple task. Well, in any case, it can be, the speed here can be increased. So if the platform does not start the pipeline, for example, we can simply reload a page, go to My Pipeline section, which includes all the information about pipelines, select our pipeline, and then press Run Pipeline again. So after the pipeline is started, we can simply monitor the progress either in the My Pipeline section or just looking at one pipeline that we have created. But if there are if the number of genes of interest, for example, number of genes identified as differentially expressed genes, number of genes with good adjusted p-value and good fold change is relatively large, it's very hard to make certain conclusions just looking at annotations of individual genes. So what are the standard steps of further analysis of lists of differentially expressed genes? There are at least two options. The first option is pathway or gene enrichment analysis based simply on the list of gene names. This very simple analysis can be performed online, for example, using a standard tool, David, for enrichment analysis, simple but widely used, called David. It's not the unique tool, but it's one of the most popular tools for this type of a job. So if we have certain differentially expressed genes, for example, let me take HR output as an example, and let me again take genes with relatively small false discovery rate, relatively small adjusted p-values, so FDR lower than, for example, 0.05, and log fold change, for example, higher than 1. So I have here more than 700 genes, and it's really infeasible to look through all these genes manually. But we can copy all these genes. We can simply copy all these genes and try to see whether many of these genes belongs to the same pathway or belong to the same set of genes with the same function. So, for example, we can go directly to required site, or if we don't remember its name, just find it there. Words David Enrichment. And the usage here is really simple. We paste our list. So, let me just show it a bit slower. We start our analysis. So we press start. We paste our list to the box with the title paste a list. We select the type of our identifiers. In our case, it's ensemble identifiers, ensemble gene IDs. We set an option that is a gene list of interest and simply submit this list. So it's simpler to do it in the previous version of David. However, the new version also works good, but previous version, David 6.7, is sometimes more stable. So you can use any of the options. So in case of the previous version, we'll get slightly different enriched categories. So again, we set identifier type to ensemble gene ID. We tell that it's a gene list. We submit this gene list. Well, it says that many identifiers are unknown to the system, and we'll currently see what's the reason. What is the reason here? So in our case, David says that it cannot process all our identifiers. Just a moment. So let me open it. What's the reason? The reason is, in our case, in structure of names. Ensemble identifiers have version at the end, so the simple attempt, at least to correct the situation that is really frequent, is to try to cut this version, so the number that follows dot, well, for example, we can say replace and replace all the text that goes after dot, including dot, with an empty set. So here I did it, and we can try to supply this shortened list of ensemble identifiers to David again. So start analysis. So probably it's the only problem with identifiers we'll have today. We set identifier ID to ensemble gene ID. We set the type of the gene list to the main one. We submit this list. And here, after removing version, we see that there is no problem with conversion, and we can select organism, in our case, Homo sapiens. So it's a standard problem with various gene identifiers we have in bioinformatics, and some systems have automatic conversion tools. Some work only with ensemble IDs, some work with only entries IDs, some work only with gene symbols. So there's a certain mass with these identifiers, but unfortunately it's standard for working with different types of gene identifiers here. And what David does is finding enriched categories. We can look at these categories at the just pressing at, for example, functional annotation charts. We see the name of categories, and the main parameter here is adjusted p-value. Here we see so-called Benjamini-Horbeck or Benjamini-corrected p-values. So the last column contains adjusted p-values, which tell us in which functional categories, in which pathways, well, there's a statistical enrichment with the genes from our list. So this type of pathway analysis is really frequent, but it is too simple for many cases, as this type of analysis ignores the majority of information we have after differential expression analysis. Here we simply ignored fold changes. Here we simply ignored p-values. We ignored even the directions of changes of genes. We set hard thresholds. So if gene satisfies these thresholds, we included it into the list. If gene was very close but was not inside the zone, it was completely excluded from the list. That is why setting slightly different thresholds can give us slightly different gene lists and different results of gene enrichment analysis. The second type of the analysis works not only with gene names or gene titles, gene identifiers, but it takes additional information we have after differential expression analysis. I mean information related to, for example, fold change or other characteristics that represent the rate of differential expression. So in order to test this type of the differential expression analysis, let us create and run one more pipeline and then discuss what's going on there. So we perform differential expression analysis. Well, at least we discussed a pipeline that can simply do this job. And after this pipeline, we had logarithmic fold changes for each gene. These logarithmic fold changes, along with all other information, are stored, for example, for the sec2 file in main file subfolder, the file called dsec2 full out and not .txt. So what information is contained here? It's a simple output of dsec2, additionally annotated with gene symbols. Gene symbols could be added via annotation, simple annotation tool we just discussed. So let's go back to one step and look at the progress of our pipeline we started. So I simply reload the page to see the current state, and I see that my pipeline, the previous pipeline I started, is done. The main output for this pipeline are stored in download pipeline output files section. And if we uploaded a file associated with 15 genes, our main output will be the file with the same name but with the suffix out. So what's there in this file? Again, I open it in Excel to have clear visualization. The first columns are the columns we had in the initial file, but then for each line, the model adds gene symbol, gene description, available gene ontology categories, and all this supporting information. So there's absolutely no need in manual addition of gene symbols when we need them. Annotation model simply does this job. And so this annotation model was applied to our output of differential expression analysis, and gene names were added simply because certain gene set enrichment analysis tools work perfectly well with gene symbols, but not with ensemble gene identifiers. So in our case, we have a complete table that particularly has a rate of differential expression measured by log 2-fold change for each gene symbol. And we can perform the identification of pathways or gene sets enriched by differentially expressed genes without setting any thresholds and including all the genes into the input data, and the model itself looks at the value of differential expression characteristic, for example, log 2-fold change, and makes a decision whether the differential expression is high or low simply in comparison with other values we have. In order to create a pipeline which performs these analyses, we also need to go to the utilities section and upload an input file. In our case, for example, if we're dealing with DSEC2 differential expression analysis, DSEC2 full out annotation file. So it may take some time for the uploading. We go on, we start our pipeline, we select, in this case, human gauge model, which is a simple application of gauge enrichment analysis to humans. And let us look at parameters we have here. So, and simultaneously look at the input file. Our input table has headers for columns. That is why we set column header present to yes. This model deals with two types of gene IDs, gene symbols and address gene identifiers. Gene symbols are specified in the second column, and that is why we set the column with gene IDs to 2, as ensemble IDs are unacceptable. Logarithmic fold changes, our characteristic of differential expression, are stored in the fourth column. That is why we set column with gene differential expression measured to 4. Then we select statistical model for the analysis. For logarithmic fold change as a characteristic of differential expression level, we generally select parametric model. For example, FDR, that is adjusted p-value, as a characteristic, we generally select non-parametric model. Then we select a set of pathways or gene sets analyzed in this analysis. For example, KEGG MET-SIG is a set of KEGG metastatic and signaling pathways. We select genes we consider only upregulated, only downregulated, upregulated and downregulated considered both but separately, or considered in combination. And we additionally set a threshold for visualization. So for all pathways that will be enriched with statistical significance with FDR lower than 0, in our case 0.1, additional plots will be drawn in case of using KEGG pathways. So we set these options, we complete creation of our pipeline, for example, human gauge test, and run it. So our pipeline is started, and similarly to the David case, we'll have here enriched pathways. We'll see it in a while. But here we do not need to set any specific thresholds. Here we use, in fact, complete information that we have after differential expression analysis. And so the results are believed to be more biologically relevant. So soon we'll see the output of this pipeline. It takes some time, as the input includes, as we saw, data for thousands and tens of thousands of genes. And after having the result for this pipeline, in fact, we'll complete the discussion related to standard differential expression analysis of RNA sequencing data. So the analysis standard for many experimental setups. Just to summarize it, standardly, we simply create a pipeline which in this platform includes preprocessing of sequencing data, mapping, quantification of expression level of genes, and identification of differentially expressed genes by computing fold changes and, which is much harder, statistical significance of the difference for each gene. If we deal with essential number of differentially expressed genes, then we might want to have a deeper insight into the biology of the whole set of differentially expressed genes, and it can be done by gene set or pathway enrichment analysis, which identifies the function, the pathway group of known group of genes enriched by genes identified as differentially expressed in our experiment. So we'll see what processes are different for our compared group of samples. For this type of the analysis, we initially need to know the groups. So if we have several groups, in the simplest case 2 groups, we can do all the steps, we can identify differentially expressed genes, we can perform gene enrichment analysis, and to have its result. So generally it takes several minutes. At this point, let us have a short break and discuss questions if required, and after this break we'll look at the output, and the next sections of our today workshop hopefully will be more dynamical. We'll run more pipelines, we'll discuss more things in the next section associated with the unsupervised analysis type, where we do not know initially what are the subgroups, and when we even do not know whether these subgroups exist or not. So please feel free to ask questions. And
Thank you, Dr. Berendenko. I hope the workshop is going good, and I'm repeating my sentence again. If anyone has any question, please feel free to reach out to us through a chat box that we have on our So
let us simply give some time for this question. In any case, for example, have a 5 minutes or 10 minutes break. If you wish, then discuss these questions if there will be any. Hopefully there will, and then go on to our machine learning analysis. As I told, hopefully it will be more dynamic.
yes, sure.
So, regarding annotation, no.
Yeah.
Yes.
Hello, Professor. Regarding annotation, after doing the DE-seq2 analysis, is the process was automated to assign the annotation, or we need to use David and other applications separately to get the analysis annotation reports?
Okay, sorry, it's audio works not perfectly well. So Hugh, as you are just closer to the microphone, could you please repeat the question?
Yeah, sure. Just a second.
Okay, sorry.
Yeah, no, just a second. Yeah.
Sorry for this technical problem.
Yeah, no, it's coming close to the mic.
Yeah, this is the question,
Right.
Professor.
Is it audible now, Professor?
Yeah, now sounds fine.
So this is the question, Professor, the doubt that I got from our pipeline. After performing DE-seq2 analysis, for annotation of the genes that were identified to be differentially expressed, should it be done through David out of our pipeline, or something that anything there in our pipeline that can even do the gene annotation process? So might be I missed some point or anything like that. That's why I want to confirm once.
Thank you very much for this question. So after performing any kind of analysis aimed at identification of individual differentially expressed genes, DE-seq2 or HR or something different, there are multiple options for gene set enrichment analysis. David is one of the options. Another option is, for example, gauge. Well, so the number of tools is really huge, not only two. But David analysis should be performed at their website, which is also very simple. But alternative method, gauge, that also performs the same pathway or gene set enrichment analysis, just in using a different and more complicated statistical model, is performed inside the same platform. So it's performed inside the same platform, and it also provides us the list of differentially of pathways, for example, enriched by our differentially expressed genes, and also illustrations of genes, of the map of genes in these pathways that have this differential expression. For example, currently, when we have a break, let us look at the progress we have here. So, yeah, probably you also have the same output if you started the pipeline. So what is the result here? Generally, just to avoid downloading too many files, all the output in the platform for the gauge gene set enrichment analysis are put into one archive. Inside this archive, what we'll have, just a second. So here's this archive. I just unzip it somewhere. So what we have here. First, just similarly to the output of David, for example, and other tools, we have a list of analyzed pathways. So if we specified that the list is a list of KEGG metabolic and signaling pathways, then we have here all metabolic and signaling pathways. And the main value we have here, just similarly to the adjusted p-value we saw in the David's case, is, well, theoretically or statistically the same. I mean, also adjusted p-value, here it is called q-value, for each pathway. So we see which pathways have high level of enrichment, that is p-value, probability of observing the same thing is low in case of no enrichment, and which pathways are not involved in the process, which pathways do not have signs of differences in expression levels between the compared groups. Again, the standard threshold is 0.05. It means that if adjusted p-value is higher than these conventional thresholds, then generally it's hard to say that the pathway differs between the compared groups. And for all the groups, all the pathways with high level of differential expression, in our case, as you remember, we set the threshold equal to 0.1 in parameters. We also have a visualization, a standard visualization. So, for example, the most differentially expressed pathways is related to focal adhesion and has an identifier 04510. So for this set and for all, and for other different pathways enriched by differentially expressed genes, we have a visualization. We have a visualization here. So we simply find a file with the required identifier and see which players are involved in these pathways and which players in these pathways are downregulated. So these players are colored with green, or upregulated. These players are colored with red. Additionally, this output contains an R script that performed all the analysis using the gauge model, and this R script can be naturally adopted for other needs. For example, slightly different visualization can be performed, so there's no need in preparing an independent script that performs this type of analysis. If some additional information is required, if a slightly different visualization is required, these R scripts can be simply corrected and run in any machine, including Windows machine with RStudio, Linux machine, or something like that. So this adaptation, simple adaptation, can be much faster in comparison with preparing a new R script. So great, probably if there are no, yeah, there are some. Great, thanks. Okay. Yeah, unfortunately, as far as I see.
Thank you.
Thanks, Ahil.
Yep. Sorry.
So, yeah, now we're both unmuted. So I'll be glad at least to try to answer one more question and then one more.
Sure. Any more questions?
It's a quick question, but what is your preferences between TPM and FPKM?
Okay, again, thanks for this very good question. From the formal point of view, if we analyze one sample, the difference is minor. For each individual sample, these measures are proportional. So there's always a multiplier, a constant that can help us to go from FPKM to TPM and to the opposite side, but these constants are different for different samples. The methodological difference between these measures is the following. FPKM has no natural biological interpretation. We understand perfectly well the reason of the definition, so we have to perform scaling by gene length, we have to perform scaling by the number of reads just to reduce the influence of these two types, these two differences in gene length and in sequencing depth on the result. But still, if we have FPKM equal, for example, to 10 or 100 or anything else, this value is hardly interpretable. TPM has a natural interpretation as its number of transcripts per million. So if a TPM measure equals, for example, 100 for a gene, it means that if you randomly select 1 million of transcripts, 1 million of messenger RNA molecules in your sample, approximately 100 of these 1 million will be transcripts associated with these genes. So in fact, TPM gives us relative expression level of each gene relative with respect to the total expression level, to the total number of transcripts in a sample. So the methodological difference is in interpretability. Additionally, there are some manuscripts that say that TPM has a bit higher reproducibility in comparison with FPKM. So if we perform the same experiment, for example, in terms of biological replicates, then for TPMs, the results will be more reproducible, they will be closer for different replicates in comparison with FPKM. But it's just a statement of a few manuscripts that dealt with these measures. So it's one of the opinions, but the opinion, well, explained with good arguments, good experimental arguments in a number of manuscripts, a number of publications. So great.
Thank you.
Great. So let's go on further just in a minute and two, as we've got unsupervised and supervised analysis for the discussion today, and hopefully more questions. So 1 or 2 minutes for break, and then we'll proceed with the unsupervised analysis. меня камера. Аудитория, этот слева посадился, что
А где все люди?
да, там вообще бывает часто. Ну, единственное, да, я вообще не вижу, брат связи никакой человек, но
А, они точно есть онлайн.
не знаю, может, нет. Ты не знаешь, да? Поэтому. А, это еще часик?
Да, можете
Ну,
ничего.
сейчас покажем.
Ты ли ваша комната?
Если надо.
Я думала, вопросов не будет.
Okay, so please let me know when we can go on further.
I guess we are having some questions over the chat, so probably we can start after a minute. I guess we can proceed, Dr. Ganatenko.
Great,
We can proceed.
great.
Yep.
Okay. So, as we discussed, for the differential expression analysis, we just please let me know is the correct screen is on now in the broadcasting. If you see the presentation, just the title of
Yeah,
the third part.
yeah, it's part 3, unsupervised analysis. That's what we are
Great.
seeing right now.
Great, great. So let us go on.
Sure.
And let us imagine a situation when initially we do not know the groups inside our set of samples, and it is a very standard situation in case of analysis of at least some pathologies, at least some diseases. And as an example today, we'll use a set of breast cancer associated cell lines. Initially, we know nothing about these cell lines. It may, moreover, I think that many of us do not know details of this pathology, and so initially we at least might think that all samples, all in this case cell lines associated with breast cancer, are somehow similar to each other. At the same time, for some pathologies, different subtypes exist, and the existence of subtypes allows optimizing therapeutical different drug strategies for these subclasses. If patients are different, if patients can be divided into these subclasses, possibly different treatment strategies are optimal for these subclasses. So what we initially have for this analysis is raw quantification, for example, in this case measured by log2 scaled FPKMs again, for about 50 breast cancer samples. This data is stored in the main file subfolder, the file cell lines expression data.txt. What we have here is expression levels of approximately 7,000 of genes for 52 samples. We reduce the number of genes, keeping only genes with sufficiently high expression level, just to slightly improve the analysis speed, but in any case, these analyses are fast. So it's very hard to analyze subgroups looking at tables, but there's a simple tool for the analysis of existence of subgroups called principal component analysis. From the mathematical point of view, this method simply selects a linear subspace which optimally approximates our initial data. For visualization, the standard dimension is simply 2, as generally we have two-dimensional plots. The idea behind the principal component analysis can be informally described as follows. So imagine that we have an object, for example, a pen, and I have a 3D pen, but what you see is a simple 2D plane projection of this pen. So if I select a poor plane, a poor projection, a poor direction of a projection, as for example I do now, you have almost no information about an object. But if I select a different projection plane, you see as much as possible for this object. You can easily identify the object type. You can easily understand the object details. So there are more informative and less informative planes with respect to the projection on this plane. So again, here you see almost nothing out of the object. Here you see an object, well, much better. The same thing is related to the analysis of samples, which are initially points in a space with number of dimensions equal to the number of analyzed features, genes, for example, but can be projected even on a 2D plane. In order to see the result of this projection, let us create one more very simple pipeline. So let us go to the areas of analysis. Unsupervised analysis. Upload these files we're talking about. Upload the file we're talking about. The file cell lines expression data.txt. Cell lines expression data.txt. And having this file uploaded, we again create a very simple, very small pipeline, which includes only one module for principal component analysis, plus drawing the resulting plots. Again, here the default options are perfectly okay. We select the number of principal components analyzed, we select scaling and centering parameters, and transpose argument tells what goes by columns and what goes by rows, samples or features. Here again, as I told, the default options are Okay, so we complete this pipeline, give it a name, for example, PCA test, and run it. So the pipeline is really computationally simple, so it won't take long to have the result. So just for starting the process, for switching between start module, main module of the pipeline, and the last module, it may take certain time, but the time won't be too long, and we'll see the result. So initially, as we discussed, we deal with samples associated with just one pathology, and so we may expect to have one group of samples with no clear differences between any subsets. So, okay, we'll have the result soon, but in order to save time, let me show you the resulting plot in the screen now, and then we'll see that the pipeline result will be just the same. Here's the resulting plot, and in this plot, we see perfectly well that samples are separated in at least two groups. Samples are separated in at least two groups. And interestingly, for breast cancer, indeed, subtypes exist. These subtypes indeed differ in optimal treatment strategies, and the authors of the manuscript, which provided the data set we're using, annotated these samples using the division into four subtypes: luminal, basal, Claudian low, and normal-like. So the number of subtypes is 4, but clearly we see here only 2 groups. If we look at samples associated with each subtype in this plot, we'd have the plot you see now. We could see, and we'll run this pipeline very soon, that one subtype, luminal, is gathered to the left of our figure, and 3 other subtypes, Claudian low, basal, normal-like, are grouped in the right part of the figure of the principal component plot. So principal component 1 versus principal component 2. Thus, the difference between these 3 subtypes is much smaller than the difference between these subtypes and the luminal subtype. So let us take a look here. Indeed, my pipeline is finished. Hopefully yours is also completed. And what we have in the output? Again, additionally, we have certain R script with additional information that can be used if a different visualization is required, or some different options are desirable which cannot be selected via the general platform interface. But the main output file is the pcaplots.pdf. So, and in this file, we see the plot we just saw in the presentation, and additionally, we can see the location of samples in plots associated with other principal components. As we specified number of analyzed principal components equal to 3, we'll have all the plots with components 1, 2, and 3. So we'll also have a plot for component 2 versus component 3. So the question is whether we can reduce the number of genes and still have a very similar information about groups of samples, about locations of samples, or have even better information about samples. So we might think that the reduction of the considered number of genes is not desirable, as more data we have, more conclusions we can draw out of this data. However, the majority of data we're looking at now is a simple noise. The majority of genes potentially have no interconnection with the question of interest. So reducing number of genes can potentially increase signal-to-noise ratio and can potentially give us more data, more information, more results in comparison with the analysis of the complete set of genes. And indeed, let us look at what's happening if we reduce our focus from 7,000 genes to just 15 specifically preselected genes. So let us construct one more principal component analysis plot. Let us go again to the section with unsupervised analysis, and this time upload a file which contains information only for 15 genes, cell lines, 15 genes marked .txt. So file cell lines 15 genes marked .txt. Here it is. And just repeat creation of the same pipeline. Just repeat creation of the same pipeline. We leave all the options in their default values. So give a name to the pipeline and start it. If we look inside the file we uploaded to the system, we see information just for 15 genes, and additionally for each sample a group is specified. This information is used by the plotting module to show the location of each group in the principal component analysis plots. So just samples associated with the same group, in our case subtype of cancer, will have the same colors, will have the same color. So this pipeline is also relatively fast. So let's just wait for a minute and we'll have the result of this pipeline available for my version and hopefully for the ones you're also starting. It indeed does not take long. And while we're waiting, let me again pay your attention to the fact that principal component analysis is a powerful method, but still the final examination of results is performed, well, intuitively, is performed by a visual inspection of resulting plots. So we can see certain separation into clusters. We have our intuition to be used to see the groups in these plots if initially we know nothing about these groups. So principal component analysis is primarily a tool for visualization, but not for clustering. And clustering is a task of dividing all the samples we have into the subgroups in which similar samples are grouped in one cluster, and dissimilar samples are generally located in different clusters. So again, my principal component analysis for only 15 genes is finished. Again, hopefully yours are also finished. And here, let us take a look at plots we have. We see, as we discussed, that the reduction genes indeed improved our signal, at least if we discuss separation by subclasses. Just automated analysis of these 15 genes, better separated subclasses, but still, as you see here, normal-like samples and basal samples at least look similar, at least have similar locations. But here, if we see no colors, we may see a different number of clusters. For example, personally for me, in this figure, I see 3 clusters. The left one, the top one, and the bottom one. In order to have formal answer in the problem of clustering, in the problem of division of samples into subgroups, we can try to use a model that specifically performs automated clustering. There are, again, multiple methods for automated clustering. Well, probably the simplest ones are hierarchical clustering and k-means, but let us hierarchical clustering using just the same input data. Let us have a look at one more pipeline. Let us create one more pipeline in the same section, unsupervised analysis. Let us upload the file cell lines 15 genes. It contains expression levels for the same 15 genes, but does not contain marks, labels of initial classes. And create a pipeline with one model, H-class model, that performs hierarchical clustering. Here, we might want to change a number of parameters. First, let me set number of clusters, but if you like 5 more, if you like 3 more, please feel free to set other values. And let us also change type. We'll discuss the meaning of these parameters shortly. For example, towards D2. So linkage type was set toward D2, and number of clusters was set to 4. So that's it. So that's it. And while the pipeline is running, let me explain briefly what is the logic behind hierarchical clustering. So our task is to divide samples, which are here represented by dots, into groups in such a way that similar samples are grouped in the same cluster and dissimilar are not grouped. The problem is that initially we know nothing about the number of clusters. So we can use a bottom-up, bottom-top strategy and start with a situation with a state in which each sample is a separate cluster. Then we simply find 2 samples that are most similar to each other, and the similarity is determined by the matrix, the distance function we specify. And further on in the procedure of bottom-up hierarchical clustering, we treat this pair of samples as one quasi-sample. So these samples are united into one quasi-sample. Then we find the next pair of samples or quasi-samples for which the distance is the lowest one and unite them and continue this process. But in this continuation, we need to define the distance not only between individual samples, but also between groups of samples, between our quasi-samples. Definitely, there are many very different ways of defining this distance between sets of samples, between quasi-samples. It can be mean distance, it can be minimum distance, maximum distance, median distance. Various definitions are possible here. And this linkage type parameter, in fact, specifies this option of hierarchical clustering, how to calculate the distance between quasi-samples but not individual samples. So this procedure is continued. At each stage, the number of groups is reduced by one by uniting two groups into one. And finally, at the end of this procedure, we have all samples united into one group. This visualization provides very little information, but much more natural visualization is standardly used for hierarchical clustering. So if we look at the output of our pipeline, again stored in the download pipeline output files, we see a PDF plot here. We see a PDF plot here in which samples are enumerated, are listed in the bottom, and then links show the order in which samples or quasi-samples were united. The height of the link is a measure, so the height of the link shows the dissimilarity between clusters while uniting. So this figure obviously shows that all our data can be naturally divided into two clusters. And if we just cut this tree at the height of, for example, 40, we'll have a separation of all the samples into two well-defined clusters. But potentially, we can further reduce our cutoff level and have higher number of clusters. So if we reduce our cutoff level to the value of, for example, 25, we'll have one group of cluster, another group of samples, sorry, one group of samples, one cluster, another cluster, another group of samples, and one more cluster. So the next reduction of height will further increase the number of clusters. And surprisingly, just a moment, surprisingly, in our case, these four clusters just perfectly coincide with samples associated with each subtype of breast cancer. Moreover, you see here that if we further reduce the cutoff height, we'll have the luminal cluster divided into two subgroups. And indeed, clinical data exists, and indeed, in standard classification of breast cancer samples of breast cancer patients into subtypes, luminal subtype is further subdivided into two groups, luminal A and luminal B. So what we see here in this simple example, we see at least two facts. The first fact is the following. Even knowing nothing about existence of different subclasses, existence of different subgroups, we can identify, we can discover this existence, this separability into subgroups by unsupervised analysis. It can be performed, for example, by a simple application of principal component analysis and visual inspection of plots, or by hierarchical clustering and again inspection of dendrograms, which clearly shows the number of evident clusters. The second fact we saw is that sometimes having smaller information is more efficient than having larger information. For example, if we're looking at certain treasures and have, well, thousands of maps, out of which only one or two has the location of these treasures, we'll have a lot of problems and we'll have to spend a lot of time looking at the required map and then looking at treasures. If we have much smaller number of maps, but these maps still include the information we need, our procedure for finding will be more efficient. The same thing is here in analyzing genes. If we reduce the number of genes, but simultaneously increase an analog of signal-to-noise ratio, then potentially we'll have more information out of this analysis, then potentially we'll have a much clearer figure of what's happening. Definitely, the problem is in the optimal selection of these genes. So currently, it's not clear how these 15 genes we're looking at were selected, but they let me just tell you that we were also selected automatically. So now initial biological, in fact, no initial biological insight was used for this selection. In many cases, it is an iterative procedure. We perform clustering based on the complete set of genes. We see that samples can be divided into clusters, and we select genes that differentiate that clusters as much as possible. For example, using a conventional differential expression analysis we discussed in the first part of our workshop. Then we limit our focus to this much smaller set of genes and clustering and have slightly different, well, optimally a bit better group of samples as we're focused on the genes identified in a way as informative ones. So for these new groups, for these new clusters, we repeat the analysis, we repeat the selection of potentially most informative genes, we then go back to clustering, and using these iterations, we potentially get better and better vision, get a better and better idea of clusters we have in our data. Just to see that there's indeed no technical difficulty in these types of the analysis, let us try to test the skills using another example of the data. So in the set of files we all have, there's a subfolder called hands-on. And in this hands-on subfolder, we have a table with expressions. For the simplicity, we left only the same 15 genes, but the number of samples here is relatively high. We have 40 samples here, and the structure of the sample space is the following. Some samples here originated from human patients with breast cancer, and some samples were taken out of the mice model, so-called patient-derived xenograft model, in which human tumor is implanted into a mouse and then grows further into the microenvironment related to a mouse. Clearly, microenvironments for humans and mice are essentially different, and obviously samples originated from humans and mice should be significantly different, even in spite of the fact that these samples are all human tumor samples and represented the same pathology. So the task I want to discuss with you is the following. Can we subdivide all these 40 samples into groups and to understand which samples are grouped together as potentially human samples and which samples grouped together as potentially mice samples? So we do not need to hypothesize which group is a human group and which group is a mice group, but we need to try to divide these samples into two groups, humans versus mice, knowing nothing more about the biology and about the clinical features behind this data set. So how can we go further and how can we find this division? So let me give you just a few minutes for this task. Obviously, we do not have a lot of tools here, but I hope that you'll find even the small set of tools we discussed today sufficient for this hands-on task. So, for example, let us try to solve this task in 5 or 7 minutes, and then we'll discuss the result.
If there are any questions, please feel free to chat with us over the chat box present on our website.
Okay, so one option we can do here is obvious. So in fact, both are obvious. We can do either principal component analysis or hierarchical clustering. So let me just try to start hierarchical clustering while you also try to perform and try to test your own ideas. I create an unsupervised analysis pipeline. I simply upload the file I'm interested in. I simply take this file out of the hands-on folder. I used hierarchical clustering, for example, setting the same linkage type, and as we expect two clusters, I set number of clusters to 2, and simply run this pipeline. Hands-on H class, for example. So hopefully, if the sets are indeed very different, then we'll really have a clear separation. And while it runs, I also create a principal component analysis pipeline with just the same file. So principal component analysis, for example, with the default options, and let me then test what will be in the plot. So here it is, and so while waiting for the results, please, as usual, feel free to ask questions. Well, as we might think that we currently have a short break, and after this break, we'll continue and have a very short but hopefully informative discussion of the supervised statement of the analysis, the discussion that, along with standard differential expression analysis, we can also efficiently solve the problem of classification, the problem of construction, similar to the problem solved by various gene signatures. So if you have questions, please feel free to ask.
Thank you, Dr. Kiranenko. And yeah, feel free to ask any questions if you have. And our participants, I guess you're doing quite good.
Okay. Okay. So let me have a look. Let us all have a look at the output we have after the hierarchical clustering pipeline. So first, we can look just at the same dendrogram at the PDF file, and indeed we see the clear separation into two clusters, the left cluster and the right cluster. If we want to see the cluster labels associated with each sample for the cutoff height that leads to 2 clusters? Well, we can simply look at the file that's also contained in the output. I'm speaking about 2 clusters, as while running the pipeline, I specified the estimated number of clusters equal to 2. If I specify this number equal to 3, then I'll have cluster IDs for the cutoff height associated with the level that leads to 3 clusters. So here we have, for example, a file which name includes cluster only suffix. And if we look at the file, we'll see sample IDs and cluster IDs. So indeed, here in this very simple case of the analysis, we have perfect separation. All samples which identifiers finish with even numbers, are mice samples. As far as I remember, mice samples. All samples which identifiers finish with odd numbers, 91, 83, 325, and so on, are human samples. So hierarchical clustering provided us this division very easily. Moreover, if we look at this dendrogram, we see that there's a clear separation of the PDX of the mouse-originated samples into 2 groups. And indeed, in this case, all human samples were taken from patients that had the same subtype of breast cancer. And for the mice case, 2 subtypes of cancer were included into the analysis, luminal and basal. And this division, this obviously seen division, simply corresponds to the division into this luminal subtype and basal subtype. Again, initially, we knew nothing about this subdivision, but the analysis shows us this subdivision perfectly well. For the principal component analysis, the result can potentially be not so obvious from the point of view of visualization, but let us see it. So here are the plots. And again, we see here, well, we potentially see here 2 groups of samples, left one and right one. Probably the bordering line, the separation line, is a diagonal one. So, and indeed, human samples are located in one semiplane with respect to this intuitively seen line, and mice samples are located in the other plane. Having already understanding of this division into subgroups, a natural question is the following: can we attribute a new sample to one of the subgroups? For example, for breast cancer, our unsupervised analysis revealed the presence of the subgroups. We have certain insights into the therapy strategy. At least we might have these insights, and we have a new patient. The question is whether we can associate this new patient with one subtype identified at our previous stage, identified at our previous step. And this is just a standard problem statement for the supervised analysis. So the input data here is a learning set which includes samples with known label classes. So after unsupervised analysis, after some discussions And scientific research, some clinical research, we already know subclasses, we already know subtypes, and we have sufficient number of samples, we have sufficient number of examples associated with each subtype. The question is how to identify a subtype for a novel patient. It can be done through various supervised methods. One of the methods that can be simply described is so-called spot vector machine. So a simple description here, not universal one, is the following. Imagine that green circles and red squares represent patients from two different subtypes in our plane. And imagine that there's a perfect linear separability. If there's a perfect linear separability, then there are many lines that perfectly separate the sets. And a natural question is, which line is an optimal one? Which line is an optimal one? A natural answer provided by Vapnik and Chervonenkins in the 60s and 70s is that the optimal line is the line which provides the separation with as much width of separating space as possible. When there is no perfect separability, we can introduce an optimized functional as a mixture of the width of this separating space and the penalty of being located into incorrect part of the plane, or 3D space if we have 3 features, or ND space if we're working with N features, for example, N genes. So in this case, penalty will be additionally given for 2 red samples, 2 green samples, and the balance between width and penalty will be determined by the parameter. So having the learning set, we have this separating line. Again, this separating line is determined based on the learning set. And then, if we have a new sample, we can look whether it looks like a green sample, like a sample from one group, or like a sample from another group. And to answer this question, it's sufficient to know this separating line. If a new sample lies in one semiplane, in a semiplane associated with green samples, this sample is attributed to the green group. And in the other case, this sample is attributed to the red group. Definitely, it's just one of the rough examples. There are many different types for classifier construction. Many of them are very dissimilar, well, random forest, for example. But in any case, the idea is that having the learning set, we try to find the rule of attributing every new sample with arbitrary features to one of the classes. And in our platform, there are multiple tools which all have their peculiar features. For example, quadratic discriminant analysis cannot be applied in case when the number of features is larger than the number of samples, or in other words, when the number of samples is insufficient. In any case, there is always a choice. At least 4 methods can be applied even in case of small number of samples. Another question is the efficiency of this application. So what example can show us the application of these methods? We already have these breast cancer cell lines divided into 4 types. And this information can be used as an input for constructing, for tuning a classifier. So we initially specify which subtype is, which sample is attributed to which subtype. So it is a learning set. And we have a set of new samples for which we know nothing. And we want to attribute them to one subclass or to another subclass. In this case, you see in these files that the data stored is just identical, but definitely it's not normal for the real situation when we use a set for learning and then want to use the same set for testing. We want to apply our classifiers to novel samples. So, to make the situation more realistic, let us remove a number of samples from our training set. Well, for example, as an example, let me remove the sample 600 MPE from our training set. If you try to perform your own experiments, you're welcome, and please feel free to remove any others samples, but I remember this sample and I remember that it is luminal. And for example, let me remove HCC 1143, a basal sample, and save the result. So these samples, two samples, are removed from our training set. There are no more information related to these samples, and let me take only these two samples as new ones. So in the set of new samples, I keep only the sample 600 MPE, and I keep the sample, I'm looking at it, here it is, HCC 1143. So here's our new test set. And the system indeed, machine learning method, supervised machine learning method, knows nothing about these samples. So in order to construct a classifier, a function which is able to deal with new samples, we need to use supervised analysis section of the platform, and the usage is quite simple. We upload training set, we upload test set. So we upload two files. We need just 2 more minutes and we'll see the results, and that will be a finish. And we select one of the machine learning methods. Well, as we discussed, there is linear discriminant analysis, for example, random forest, support vector machine, naive Bayes, for example, let it be naive Bayes. And complete the pipeline. So test naive Bayes. So this pipeline will construct a classifier using only our training set, and then apply this classifier to our new samples from our test set. Just similarly, we can construct a classifier which uses completely different mathematical model. Well, let it be, for example, random forest. For deep analysis, for deep understanding of these mathematical models, essentially more time is required for the discussion. But again, we upload these files, we tell the system to construct a classifier using random forest method, and this classifier will be then automatically applied to our new samples. While pipelines are running, let me just summarize the main points of our discussion we had today. First, in many cases, no coding skills, no statistical background, and no mathematical background is required for standard processing of RNA sequencing data, even starting from raw data. This processing standardly includes preprocessing that can be avoided without an essential effect, but still adaptive trimming is desirable, expression level quantification, and in many cases differential analysis can be performed. All this can be performed using a simple mouse interface in the server.t-bio.info platform. So for differentially expressed genes, there are multiple characteristics of differential expression. We can't look only at fold changes. For example, statistical significance is important, and quotient is important for genes with low expression. Then, having this list of differentially expressed genes, we can perform gene set enrichment analysis or pathway enrichment analysis. Again, it can be performed without coding skills. If we deal with standard setting, human gauge is one of the possibilities for this analysis, but having coding skills can help us to adapt the outputs, to adapt, for example, visualization to our needs. Next, if we deal with samples without initially known structures, if we simply quantify expression levels, unsupervised analysis methods can provide us this insight, at least can tell us whether all samples comprise one group or can be naturally subdivided into groups, into clusters. Today, we discussed only one approach to clustering, however, multiple approaches exist. And then, if we know groups, if we know which groups are associated with the question of interest, for example, with the pathology, then we can, and if we have a learning set of examples for each subgroup, we can construct classifiers using supervised machine learning analysis approaches and attribute new samples to one group or another. For example, we can look at the results of the naive bias classification, see subclasses for the test set. So I look at files with the test set suffix, and I see that 600 MP sample was classified as luminal and HCC 1143 was classified as basal. So indeed, the classifier that knew nothing initially about these samples classified these samples correctly. In a similar way, we can look at the results of random forest classifier. Again, in the set of output files, we'll have a file with a res for test set suffix, and for random forest classifier, again, we see that the classification results are correct. Definitely, we can't expect this automated method to give perfect results in all the cases, but in simple cases, their performance is quite good. So overall, today we covered initial processing of RNA sequencing data, preprocessing, expression level quantification, some conventional approaches for differential expression analysis, and discussed the statements and the ways of going through unsupervised and supervised analysis of samples aimed at classification. Thank you very much for your attention, and please feel free to ask questions. So let's try to see what are these questions, and you're welcome to have your own experiments with all these data sets, to test various methods, to compare them. So this hands-on experience you can probably have now and definitely is able to have in the future is hopefully really good and really helpful, as here all the time we worked with real data. In the majority of cases, the data originated from the Cell Lines project, so breast cancer cell line project. That's it.
Thank you. Thank you. Thanks a lot, everyone. Thanks, Dr. Garantenko. Thanks, Julia. Thanks, Jack. Thanks, Avi, and all the participants for taking all the time for this workshop. We have our basic courses on edu.t-bio.info, and we have the server access. You can try out and play around with different pipelines. The access is valid for 14 days from now. And if you have any questions, any doubts, feel free to reach out to the chat box, or you can email either one of us. And if there are any questions, feel free to reach out to us. Thank you.